Hi everyone! Today we're going to be going over a really important lab technique that's called Lac Z or beta-galactosidase staining. Just want to share that the images that are in this video are credited to the research paper Parental Olfactory Experience Influences Behavior and Neural Structure in Subsequent Generations by Diaz and Ressler, published in 2014. So what exactly is Lac Z beta-galactosidase staining? Simply put, it's a lab technique to visualize activity, much like how I'm showing you this definition. A great way to see the efficacy of this technique is by looking at an example. This image is pulled from the Diaz and Ressler paper, and here we see the image of an olfactory bulb. And in this experiment, the researchers were trying to identify neurons that expressed the M71 odor receptor, which is coded by the gene M71. And through the beta-galactose staining technique, we can see that wherever we see blue in this image of the olfactory bulb, that is where neurons that express the M71 receptor are located. And we can see the applications of this kind of technique. It allows us to make comparisons across different groups and is very helpful for gaining a deeper understanding of certain things that are being experimented on. So without going into too much of the details here, we can see that of the three categories here, the category F1 ACE M71 displays a significant amount of staining as compared to the two other groups, F1 home M71 and F1 propanol M71. And so in this case, we can see how the beta-galactosidase staining is really helpful in helping us visualize this kind of difference. So that's great and all, but how does it do that? How do we get these neurons that express this particular receptor that we're interested in? How do we get them to show the blue? In order to understand that, we need to look at the mechanism of the lac operon. So here we have an image of the lac operon in E. coli, and while there are a lot of moving parts to this mechanism, we're going to focus on the Lac Z gene, demonstrated right here. So when the operon is turned on and this DNA sequence is all transcribed into mRNA, Lac Z is going to express Lac Z. And now they have the same name, but there's an important distinction. The gene Lac Z has a lowercase l, whereas the enzyme that is coded for by the mRNA has a capital L, so that's a way you can distinguish it. That, or you can also refer to it by its other name, beta-galactosidase. And beta-galactosidase's job is to take the incoming lactose and basically break it down into glucose and galactose. So let's zoom in into what this looks like. Here again, we have that same exact Lac Z or beta-galactosidase enzyme, and we have lactose. And we call whatever enters or fits this kind of lock shape of the enzyme a substrate. So the enzyme could be the lock, and then the substrate could be a key. And so the incoming substrate comes in, takes its spot, and once it fits into its little cozy position, then the enzyme can do its job and break down the lactose into galactose and glucose. As it relates to our technique, however, instead of lactose, we have X-gal, a different kind of substrate that still goes to the Lac Z enzyme. And so when you incubate this enzyme with X-gal, this substrate, and allow it to fit into the enzyme, just like how lactose did, as a result, instead of glucose and galactose, we get this insoluble blue dye. So as it relates to our experiment, what we do is we take the Lac Z gene and integrate it into the mouse genome. We integrate it in such a way such that when the M71 gene is expressed, we also want the Lac Z gene to be expressed, so that when you incubate with X-gal and the M71 receptor is expressed, we also get the insoluble blue dye. Of course, the Lac Z gene is not specific to M71. You could apply this technique to any kind of gene that you want to be expressed or that you want to see visibly the expression of, and that would depend on how you integrate it into the mouse genome. But in this particular case, it was integrated such that the Lac Z gene would be expressed when M71 is expressed. And that is it. Thank you so much for listening and hope this was helpful.